Remember that vomiting bug on campus? People are still getting sick, so we need to work out what the bacteria responsible is. In the last lab class, we plated out bacteria from the contaminated water sample using aseptic techniques. In this lab, we'll try and work out which bacteria are present in the sample using a few different approaches. Remember, we can use a combination of different characteristics to identify bacteria. These include the morphology, i.e. the shape of the cells, colour changes of colonies grown on chromogenic agar, and selective staining techniques. In this practical, we're going to use the gram stain to identify whether our bacteria are gram-positive or gram-negative. Gram-positive bacteria have no outer membrane but have a thick layer of peptidoglycan cell wall. Gram-negatives only have a thin peptidoglycan layer but have an additional lipopolysaccharide outer membrane, so this creates an additional compartment in the cell known as the periplasm. The gram stain can be used to tell the difference between the two types of bacteria. There's another video available on the Canvas site explaining how the gram stain works that you should also watch. It will be really important to use the Bunsen burners correctly in the practical so that we're safe. You'll need to use the Bunsen burners to prepare your slimes, but you must switch the Bunsens off when you start the gram stain protocol, as the chemicals we're going to use are flammable. When you've stained your cells, you can use the compound microscope to see whether they've taken up the stain or not, i.e. tell whether they're gram-positive or gram-negative, and also to observe the shapes of the cells. As bacterial cells are only 1 or 2 micrometers across, we'll need to use the times 100 magnification and also use immersion oil to increase the resolving power of the microscope. Remember to wipe the oil off the microscope objective lenses when you're done so you don't damage them. You're also going to use your chromogenic agar plates to help you identify the bacteria. Remember, bacteria that can metabolize lactose produce the enzyme beta-galactosidase. This enzyme can cleave the artificial substrate red galactosidase, which is contained in the CUTI plates, and this will turn the colonies pink. It's therefore very easy to identify a lactose metabolizing species such as E. coli. Once you've got the results of the morphology, the staining and the chromogenic agar plates, you should be able to work out the likely species of bacteria. Note that these tests wouldn't be enough for a definite identification in a hospital lab. If you were doing this for real, you'd also want to do some sort of genetic analysis, but these techniques will give you a pretty good idea of what the species might be. The other main task in this practical is to determine how many bacteria were in that contaminated water sample to find out how heavily contaminated it is. For this, we're going to use the spread plates that contain your cereal dilution samples. You'll need to count the colonies on each plate and then do a short calculation to work out how many bacteria were contained in the original sample. There's another video on the Canvas site walking you through this calculation, which is also described in the practical handout. Before the practical, you'll need to take the pre-practical quizzes again. Again, there'll be a stage A and a stage B, and it's stage B that will count towards the marks of the module. After the practical, you'll need to complete the worksheet at the back of the practical handbook, which will include your experimental results. So I look forward to seeing you in the lab to identify what those bacteria are.